Hi, my name is Stephen Alsop, and I'm happy to share with you some of the most relevant information for success on the USMLE Step 1 exam. You will notice that the neuro section is a bit different from other sections. One of the most important differences is that anatomy and physiology are in the same section. This actually highlights a very important principle that I discussed during embryology, and that is that anatomy and physiology are intimately linked in the brain. Thus, we will always try to link anatomy to the different functions that we will discuss. So now let's start with the star of the show, which is the neuron, of course. This is where all of the action takes place. Neurons have a large cell body with many organelles, including the nucleus, the rough endoplasmic reticulum, which is the Nissl body, and then mitochondria. But of course, what makes the neuron not your typical cell is that it has dendrites and an axon that allows it to transmit signals throughout the brain. Now, the dendrites are where input is received, and you have a number of different arborizations on the dendrite that receives information through synapses. Now, information is transmitted from each dendrite down into the cell body at the axon hillock. And so you have a bunch of different signals coming in, and they're all going to be summated at the axon hillock. Once you reach a certain threshold, then you get an action potential, which will be transmitted down the axon, which may be myelinated or not. Here's the myelin, and we'll talk about that and the significance of what that means. Once the action potential travels down the axon, you get to another synapse where neurotransmitter is released to the next dendrite of a nearby neuron, and that signal is then passed on as well. Most neurons are multipolar, which means they have several dendrites as illustrated here, as well as several axons. Some neurons are bipolar, meaning they have one dendrite and one axon. Neuronal cell bodies do not regenerate, so any damage to the cell body is permanent. However, axons can regenerate at a slow rate. Any idea of how slowly neurons regenerate? The estimate is about 1 millimeter per day, as long as the cell body is not damaged. Now, following injury to an axon, a special type of degeneration occurs that starts at the point of injury and spreads distally. This is known as Wallerian degeneration. Now, the main two types of cells in the nervous system are neurons, which we've just discussed, and glial cells. Glia are the support cells of the nervous system, and astrocytes are one of the best studied of all the glial cells. Astrocytes derive their name from the Greek astron, which means star. And if you squint and look sideways, you can sort of see why this is the case. Maybe I can convince you that this sort of looks like a star. Astrocytes are a key component of the blood-brain barrier. They provide physical support to the neurons. They take up excess glutamate and GABA and are responsible for regulating local pH as well as ion balance. A major function of astrocytes is repair following trauma in a process known as gliosis. The astrocytes proliferate, swell, and undergo fibrosis, producing a glial scar in place of the damaged neurons. Astrocytes can be identified by a stain for GFAP, which stands for glial fibrillary acidic protein. In fact, this photo is of astrocytes that have been stained with a fluorescent marker for GFAP. Microglia are the macrophages of the CNS. Now, what are the function of macrophages? Well, they're important cells within the immune system that help to engulf pathogens and cellular debris. They also help to activate other immune cells. When tissues are damaged, microglial cells are upregulated and chew up on the injured cells. You can see the microglia cells here staining brown for lectin. Microglia make cytokines as well as neurotoxins. These neurotoxins can be things like nitrous oxide or glutamine, and these mediate inflammation. They're also important for restitution of the CNS environment, but like all forms of inflammation, they can cause lasting damage. The activity of microglia has been shown to be involved to some extent in the formation of beta amyloid plaques, which are seen in what major neurodegenerative disease? The answer here is Alzheimer's disease. Alright, time for a flash quiz. Which primary germ layer is the basis for microglia? The answer here is the mesoderm. Now the rest of the nervous system is derived from the neuroectoderm, but you really need to remember that microglia come from the mesoderm layer. They both start with M, so you can use that as a sort of mnemonic. Myelin is present in both the central and peripheral nervous systems. In the CNS, it is produced by oligodendrocytes, while in the PNS, it is produced by Schwann cells. We will talk about these cell types in more detail soon. Myelin dramatically increases the conduction velocity of electrical signaling, and it does that by increasing the space constant. This drawing shows the nodes of Ranvier. These are spaces that separate the different portions of the myelin sheath. This electron micrograph shows the myelin sheath circling around the axon. So this is your axon here, and multiple layers of the myelin sheath. Schwann cells myelinate axons in the peripheral nervous system. Unlike the oligodendrocytes in the CNS, which can myelinate multiple axons, Schwann cells can only myelinate one axon at a time. For step 1, you should be familiar with Schwann cells in two clinical scenarios. In the first scenario, the patient would present with muscle weakness or paralysis. The patient might have had a recent infection with CMV or Campylobacter jejuni. What disease would this be? This would be Guillain-Barré syndrome. This is due to autoimmune attack of Schwann cells. Now, molecular mimicry is what is important for this association between Campylobacter or CMV and Guillain-Barré syndrome. Molecular mimicry results when the immune response created as a result of infection is now tuned to recognize proteins on the Schwann cells. The immune system begins to attack Schwann cells, which, as we discussed, creates myelin. The lack of myelination on peripheral nerves innervating muscles manifests as muscle weakness. Now, in the second scenario, you might be given a picture like this. Now, this patient presents to your office with sensory neural hearing loss. Upon further questioning, you find out that they also have had trouble with standing from a seated position. What is the diagnosis here? The diagnosis here is a schwannoma. This is also known as an acoustic neuroma. This is a very common tumor of Schwann cells. The symptoms are the result of the fact that this tumor affects the function of the 8th cranial nerve. Another great clue is that the tumor is located at the cerebellopontine angle. You should definitely remember this association. A schwannoma leads to symptoms that are a result of the tumor affecting the 8th cranial nerve. Oligodendroglia are the major type of glial cells found in the white matter of the central nervous system. The main purpose of the oligodendroglia is to myelinate the neural axons in the CNS. Oligodendroglia can myelinate multiple axons as seen here. In fact, they can myelinate as many as 30 neurons at a time. Remember that white matter is myelinated axons while the gray matter is made up of cell bodies. In fact, myelin is what gives white matter its appearance. As you can see in this picture, this is the white matter, and then this would be an example of the gray matter. While appreciating the distinction between white and gray matter, you might also notice this huge tumor. This is an oligodendroglioma, a tumor that develops from oligodendrocytes. You'll notice that the tumor is located where one would expect to see the white matter tracks. Now, if we zoom in to a microscopic level, we might see something like this. In this image, you can see perinuclear halos and the fried egg appearance of oligodendrogliomas. We'll discuss the significance of this later in the pathology section. So here is, maybe your fried egg. That's your yolk. One thing that you want to be aware of is that oligodendroglia are destroyed in multiple sclerosis, which is seen on MRI as spots with loss of white matter. That's a commonly tested item on the boards. Okay, I think it's definitely time for a flash quiz. So, how slowly do neurons regenerate? The estimate is about 1 millimeter per day as long as the cell body is not damaged, and that's key. It's really amazing to think that all of our sensations are mediated by these small cellular receptors that are responding to mechanical stimuli. There are multiple sensory receptors underneath our skin, and they mediate touch, pain, vibration, etc. And all of these sensory receptors are activated by specific stimuli. These stimuli are then transduced into electrical signals which synapse with afferent neurons in the spinal cord. Specifically, either the dorsal column medial lemniscus pathway or the spinothalamic tract, depending of course on what the messages encode. And we'll discuss them one by one and the type of information they process. Now, nociceptors contain free nerve endings, and they are made of two different types of fibers. A delta fibers are myelinated and encode painful mechanical stimuli like a sharp pricking pain, while C fibers are unmyelinated and respond to hot, cold stimuli and blunt trauma. Now, the A delta fibers encode fast and first pain, while C fibers encode longer lasting, dull second pain. This makes sense, right? The fibers that are myelinated and thus carry the message at a faster velocity mediate the message of pain to your brain. Meissner's corpuscles are large myelinated fibers found in the dermis of hairless skin, such as the fingertips and lips. They have pretty small receptive fields, so they are mostly found where fine touch is especially good. Purkinjean corpuscles are large, myelinated fibers found in the subcutaneous layer. They primarily mediate pressure as well as vibration. Finally, Merkel's discs are large, myelinated fibers found in the skin that respond to vertical indentations onto the skin. This would be, for example, when you press one finger into the palm of another hand. Try it out. That's your Merkel's disc working. Now, Ruffini endings are a very slow adapting mechanoreceptor. They respond to sustained pressure and responsive to things like stretching of the skin. You can try that out too. When we think of a peripheral nerve, such as the radial nerve, we are really thinking of a number of different axons from neurons that are organized together. And this organization is worth knowing. The first layer is the endoneurium. This is the layer right at the nerve fibers, and you can think of this as endo meaning inner, so it's the closest layer. The next layer is the perineurium, and that peri means around, and so you can see that the perineurium kind of goes around all of the nerve fibers. And last is the epineurium. This is the dense layer that contains several bundles of nerve fibers, as well as their vascular supply, and epi of course means outer. So out of these three layers, which layer do you think is the most difficult for drugs to cross? It's actually the perineurium. You can think of it as the permeability membrane. It normally serves to protect the nerve from damage by cytokines and protons that can cause abnormal nerve conduction. Now, this property is the result of its basement membrane as well as the tight junction proteins that form the barrier between the perineurial cells. Now, while we are at it, what other structure contains tight junctions and can also be an effective barrier for treatment of brain disorders? That would be the blood-brain barrier. And so we've talked a lot about the nervous system and electrical signals within the nervous system, but of course to communicate between one neuron and another you need neurotransmitters. And prior to the 1920s, it was not known whether neurons communicated through electrical or chemical signals. The way it was discovered was actually through a pretty cool experiment by the Nobel-winning scientist Otto Loewi. Now, what Loewi did was to take two hearts and put them both in saline. He stimulated the vagus nerve of one heart electrically so that it made that heart beat at a slower rate. He then took the saline from where the vagus nerve innervated the heart and put it on the second heart. He was able to slow the second heart down. This proved that there was some soluble chemical secreted from the vagus nerve that could control heart rate. This chemical was the neurotransmitter acetylcholine. And signaling via neurotransmitters is the key mechanism by which neurons communicate with each other. There are quite a large number of neurotransmitters, and this list is certainly not all-inclusive. However, these are some of the most studied and well-known ones, so it's certainly worth memorizing. Now, step 1 will not only expect you to know these neurotransmitters, but where they are synthesized as well. It is much easier to memorize these neurotransmitters in light of their function in various diseases, as shown in this table. It is also helpful to know what normal processes they are involved in so that you are able to make educated guesses based on symptoms if you don't remember a particular disorder or the neurotransmitter that is involved. Now, one way you might be tested about neurotransmitters is through biochemistry. For example, how are dopamine and norepinephrine related to each other? Dopamine and norepinephrine are both catecholamines. Dopamine is used as the substrate for the generation of norepinephrine. The enzyme responsible for this is dopamine beta hydroxylase. These are the types of details you should know to maximize your chances of success on step 1. Now it's time for another flash quiz. A patient presents with hearing loss. On imaging, a tumor is found to be located at the cerebellopontine angle. What is at the top of your differential? So, what should be at the top of your differential is that the tumor is likely a vestibular schwannoma. This can sometimes also be known as an acoustic neuroma. This is a tumor that is made of Schwann cells. The symptoms are the result of the tumor affecting the function of cranial nerve 8. The blood-brain barrier is a tight endothelial layer that prevents most large water-soluble substances from entering the brain milieu. As I mentioned earlier, it is composed of a layer of endothelial cells along the vasculature with tight junctions. Surrounding the endothelial layer, there is a basement membrane, and astrocytes form foot processes surrounding the basement membrane in order to further support the barrier. Now, does this sound like any other filtration system in the body? Well, of course it does, or I wouldn't have asked the question. It sounds just like the glomerulus, which contains an endothelial layer, a basement membrane, and podocyte foot processes. Some substances like glucose and amino acids can enter or leave the brain through active transporters in the endothelium. Lipid-soluble substances can simply diffuse right through the membrane. This is why some drugs can enter the brain. There are also some specialized regions of the brain where there's increased permeability to the external environment. The most notable region is the area postrema. This is in the caudal wall of the fourth ventricle in the medulla. This is important because it is known as, anybody? The vomiting center. The blood-brain barrier can also be breached in several important situations, such as brain tumor, infection, or infarction, and this can lead to vasogenic edema. This will show up on imaging scans.